Okay, looking at this was not a well answered question. So how does the detergent release CNPW from cells? So you need to use this information here. It tells you that the detergent dissolves lipids. So how are you going to get where are lipids in cells? You've got the phospholipid bilayer, so that's important to start off with. The cell membranes are made from phospholipids. So what is the detergent going to do? You don't know if this carries out hydrolysis. You don't know it's going to break it down. It dissolves, so it's dissolving the phospholipid bilayer, which is the membranes. For the next one, explain how ultracentrifugation separates CNPW from other molecules. So what does ultracentrifugation do? How does it separate molecules? Well, it spins them very fast, or at high G, so it spins them very fast. You can't just say the standard answer for the method behind it, where you're separating, say, nuclei and mitochondria, because that's not what it's asking. So how does it separate molecules? First, what does a centrifuge do? It spins at high speed, and what does it separate things based on? Either the mass, the size, or the density. So too many of you went along the lines of that organelle separation, separating nuclei and mitochondria, for example. It's not really what it's asking. In this question here, we've got small interfering RNA. So small interfering RNA should straight away be thinking this is preventing translation, it's stopping mRNA getting to the ribosome, it's chopping up mRNA. One big problem with this was not relating to the question. So yes, you know what siRNA does, but related to the question. So it interferes with the expression of the CNPW gene. Okay. So, as you can see from the data, there's less of the CNPW being produced over time, and as CNPW is needed for tubulin production, then there's less tubulin production over time as well. So how would you get the marks? Well, the siRNA binds to, or destroys, the mRNA for CNPW. You can't just say siRNA destroys mRNA. You've not related that to the question. So what's then going to happen? It's going to stop that mRNA getting to the ribosome, so it's going to prevent the translation of the CNPW mRNA. Again, relating to the question. As a result of that, the amount of CNPW is going to reduce, therefore tubulin production also reduces. So make sure you use information from the question. Be specific. Right, in this question it says, well, why were the units micrograms per gram used? So why were micrograms used? Well, micrograms is micro is a small amount, so why would you use micro? So you might want to use micro to prevent there being lots of decimal places. So if you use grams, or if you used milligrams, you might need 3 decimal places before you have your first number. So micrograms, because there's very little ammonia in the soil, it avoids using lots of decimal places for each number. And then why is it per gram? Well, per gram allows comparison, because there could be different masses of soil used in each sample, so per gram allows comparison. For 4.2, it's 6.2, it's asking for the difference in the rate of breakdown. So all it's asking you to do is work out the rate for A over the first 2 days, the rate for B over the first 2 days, and then just calculate the difference in rate. So for the first 2 days, up to this point here, so the difference divided by the time, which is 2, and for A up to here, what's the difference divided by the time, which is 2. So that will give you the numbers. You take them away from each other, and that gives you the difference in rate, but then you need units, and a lot of you missed units. It asks specifically for you to put units with your answer. You cannot afford to do that in the exam. Even if you get the wrong number, you get a mark. Well, it'll help you get a mark for the units. So micrograms per gram per day. You're dividing y by x for the numbers, so you do exactly the same with the units. Micrograms per gram, day minus 1. 6.3 asks you what evidence supports the conclusion that there are different communities of bacteria in soils A and B? It's a very confusing question, but if it just asks for evidence from Figure 6, just use the evidence, just use data to illustrate your answer. So without reading the question even, you can give conclusions using the data. So you could easily say that B has the fastest, so not just faster than A, because it's faster than both lines, the fastest rate of breakdown of ammonia. You could say that A and B has got the slowest, slower than both A and B individually. A plus B has got the slowest rate of breakdown, and that'll get you 2 marks. And the third mark's for linking it to the question. This suggests that this community of bacteria at B, for example, work best by themselves, that they don't work well at other pHs. Okay, this question 6.4 is asking you really, do you understand logs of the powers of 10? So you can see from here, you've got a difference in a factor of 2, it goes from 10 to 6 to 10 to the 4, and here it goes from 10 to the 2 to 10 to the 4, so both a power of 2. But this really, 10 to the 6 is a million, and 10 to the 4 here is 10,000, so it's the difference between a million and 10,000, so that's 990,000. And for T, it goes from 100 to 10,000, so you've got a difference of 9,900. So there's a bigger difference for S, because that drop is a lot bigger, so it's not compared to this increase there. So you've got a 990,000 drop compared to a 9,900 increase. So it's checking to see if you understand what these numbers actually mean. So that's 10, 100, 1,000, 10,000, 100,000, 1 million. So the number tells you how many zeros there are after the 10. Sorry, after the 1. Okay, this question wasn't easy. How does it allow you to estimate the expression of the gene, but it doesn't allow you to work out the growth of the bacterial culture? Well, a mark I'd hopefully that you would put is the idea that they've not actually counted the number of bacteria. So how can you work out the growth of bacteria if you don't know how many there are? So that's a point that hopefully would be obvious. Other marks that perhaps you could have got is the idea that yes, you might know how much of the amylase gene is expressed, so you might know how much, but you don't know how much of that mRNA is produced, or that gene is produced by each cell. So therefore you can't work out the number of bacterial cells just from the mRNA, because they might produce different amounts each. And also you don't know if this gene's got anything to do with growth. It might have nothing to do with growth. But yeah, tricky question. And last one, maybe because you're rushing, but a lot of people got this wrong and just put flaming. You've got to read the question. So you've got sterilising bottles. So how do you sterilise the bottles before they're used? If you flame, you're only sterilising the neck, you're not sterilising inside the bottle. So you could use boiling water or steam or an autoclave or disinfectant, or you could wash it with alcohol, something like that. So it's actually going to kill all the bacteria inside the bottle, not just the neck. And so why do you do it? It's to kill the bacteria, but link it to the question. Yes, that would produce contamination. What might the other bacteria do? They might break down ammonia, which would affect your results.